All right, well, that's I'm really happy to be here to talk to you today and tell you a little bit of what we've been up to with respect to the search for life on icy ocean worlds and some experiments we've been doing to look at the viability of microorganisms and also some models we're putting together to help designing mission concept that would potentially at some point, we hope, go and visit the surface of this ocean world. So here are the great people who have been participating in this work at JPL. And I would also like to thank Wolf for inviting me to speak to you today. All right, so first, before going into the lab details and so on, why are we searching for life at ocean worlds? Well, the search for life is obviously a major goal for many scientists, many space agencies. This is something that has been going on for a very long time, but where we've been very active in it, at least since the late 60s. And it can take very different forms. So the first obvious one is observation and search directly for intelligent life. The SETI Institute has been great at that, and it's really what triggered a lot of the search for life in the astrobiology field. There are also some telescopic observations to search for signs of life and signs of habitability at particular worlds. It's something that is going on a lot in the field of exoplanets. And recently, we've been having a lot of activities with JWST and really many great discoveries. We're also sending missions in the solar system to explore planetary bodies and to search for life directly. And this is something that perhaps the Viking missions that went to Mars in the 70s have been really catalyzing and putting in the mind of people already that there might be life out there. A search for life on Mars now is taking slightly more different approach where we're searching for past life now on Mars with the latest missions. And we also can thank the contribution of field and laboratory work in the search for life in the sense that they're helping to explore the limit of life to tell us what it is about and what we can expect to find. So Mars has been a great target to search for life for a while, but now our eye is turning toward different bodies in the solar system, and namely ocean worlds. So what am I talking about? I'm talking about, for most of these ocean worlds, there are icy moons of giant planets. So for example, Enceladus, Europa, it's the one that triggered a lot of the work that we're doing now for the search for life on ocean worlds with the Galileo mission that went and observed this like great ice-covered surface, found that the worlds were geologically active, and also with some of the instrument, magnetometer in particular, found that there could be some conductive material in there that could be liquid water. So these worlds typically have a cold, icy surface because they're really far into the solar system, but they still have water. But their core is warm. So for some of these icy moons, there could be a layer of liquid in between that could be interacting with the inner core. And this liquid water is one of the reasons why searching for life there might be a great opportunity to find life in our solar system. We'll also have specific physical conditions there, namely the presence of an energy source. It can take different forms, can be radiation, but it can also be activity at the core, potentially hydrothermal vents, like this could be the case for Enceladus. And we've been exploring this world, so telescopic observations for a lot of them, but also with some missions. And what we found was a very large variety of chemicals, and some of them for some of these worlds, large organics that could be perhaps the sign that life could have developed there. And at least it would make some of these worlds potentially habitable, and they might be supporting simple life forms like microorganisms there. But if we send a mission to some of these worlds, the question is, if microorganisms make their way to the surface, there are some mechanisms for that. So from the liquid ocean, there could be plume, there could be also cryovolcanism of another source that could bring some of this material up. And if we send a mission there to detect and to search for life directly, what are we going to expect there? What do we hope to find given the harsh conditions that most of these moons are subject to? So that's one of the questions that we're trying to answer. The other relevance to the results I'm going to show you is also for planetary protection. This is not why these experiments have been designed for and what we're trying to do. I don't work in the planetary protection group, but the results that I'm going to show you could be also useful if we send a mission and if we inadvertently leave some material in there and potentially some microorganisms from on Earth. Would they be able to survive at the surface of these icy ocean worlds? And would it be a problem for planetary protection? So that's kind of the work that we're doing. And when I'm talking about harsh conditions at the surface of these ocean worlds and at the icy surface, I'm talking about different kinds of radiation. Solar UV photons are going to be potentially a problem. Radiation like from charged particles like electrons, but also energetic ions will be able to make their way to the accessible surface accessible to London, likely in the millimeter centimeter range at the top surface. There are also even more energetic electrons that can make their way down to meters. So that's going to be a very active environment, and we need to worry about these degradation mechanism if we send a mission that would go there and detect potential biosignature or even life itself. So the conditions that we have there and that we're trying to simulate in the lab to see if the signatures would survive are namely low temperature. We're talking about ice. We're very far in the solar system. Typically 100 Kelvin is what we're looking at, plus minus 50 K. The intense weathering from the radiation, I'm going to talk here a lot about solar UV photons. I'm going to talk a little bit about charged particles as well. I'm not going to talk about micrometeorite, but that's something that would be really interesting to look at. We're also talking about chemicals in the ice. So if you have radiation that would degrade potential signatures of life or life itself, it would also interact with the ice that surrounds it. And in the case, for example, of charged particles, you may create very harmful radicals, very harmful ions that can interact with the signatures we're trying to look at. So that's something to think about, to consider, and that we're trying to predict to see if, yes, if there will be something to see out there if at some point we send a potential landed mission. So we decided to do experiments to first look at the viability of microorganisms. So there are many different potential biosignatures that would tell us that life would be there, but how cool would it be if we could find life itself? And there are actually some instruments and some payload for concepts that are imagined to image potential microorganisms at the surface of ocean worlds. But the question is, yeah, will they be there? Will they survive? So what we decided to do was to test that, what happens to these microorganisms in ice. And for this, we chose specific microorganisms that are bacterial spores. We chose them to provide an upper limit because on viability, because these bacterial spores are very hard, very tough. They can withstand a lot of very harsh conditions here on Earth. We find them at many places, going from glaciers, but also to deserts. And bacterial spores, well, endospores, are actually a form of vegetative cells that when it's in an environment that is that it doesn't like, that it is not happy to duplicate and to grow, it's going to turn, it's going to lose a lot of its water, it's going to form a very thick coat around it, and it's going to be tough and survive for a very long time because of these protections. They're extremely resilient, and that's why they're typically being used for planetary protection to assess the cleanliness of spacecraft and decontamination protocols. There's been a lot of experiments that have been done with respect to the viability of bacterial spores, and even in the case of space science, there's been some great experiments where these spores have been sent to the vacuum of space to see how long they would withstand the radiation, so in the near Earth orbit. There's also been a little bit of experiments done at low temperature, but actually not so much. And that's why we come in and why we decided to do this work. Namely, some experiments have been done at around 100 Kelvin, and realized that the low temperature may increase viability. So that's what I'm going to talk about also later in this talk. But we need a lot more of these experiments at lower temperature, but also for different radiation conditions. We've learned a lot since these previous experiments have been done, so I think it's time to revisit the subject. And we also need to assess the impact that ice have on the viability of microorganisms, and especially if it's going to shield them, or on the other hand, if it's going to create some harmful radicals or ions that may affect their viability. So how did we do this in the lab? Well, we try to reproduce the conditions that we found at the surface of these icy ocean worlds. So using what we use typically in astrochemistry to look at ice and to study ice, so vacuum chamber, cryostats, and radiation sources that we adapt to these vacuum chambers. That's what you can see on the right here. But instead of growing ice or looking at material, what we did was to deposit a layer of bacterial spores that I had the pleasure to grow in the lab. I'm not a biologist, so that was interesting, going back to some biology that I've seen in high school and trying to redo that at a higher level. So we grew a stock of bacterial spores, and in particular, we chose Bacillus subtilis in our case because it's really an easy microorganism. It's present everywhere, so it's really safe to work with. And we deposited it onto what you see here, these stainless steel tabs, making a layer here and trying to really stay in a submonolayer regime. So this is an SEM image of these spores into the surface, and really trying to avoid clumping. Some of these experiments at some point were done dropping droplets, but here we choose a different approach, and I will show you later why. So we added these bacterial spores, these tabs, onto the cryostats. The tip of a cryostat, we attach them. You can see a figure here with the tabs in there that are being in the vacuum chamber and are being irradiated by UV photons. So it's a mini arc UV lamp that is creating a black body that is pretty similar to the solar spectrum of the sun. We have 4 tabs here with our bacterial spores, and we also have controls there, which are not being irradiated. We cool down the spores at specific temperature. We have a cryostat where we can control the temperature all the way down to 11 Kelvin. We irradiate our spores for a specific amount of time. And then we recovered them. So here's procedure using polyvinyl alcohol film. We pick up the spores, we dissolve them back into a liquid. And then what we do is that, well, we we grow them onto a petri dish and we count the survivors. So we do that for the irradiated bacterial spores. We also do that for the controls. Here's me counting old school with the manual counter. And what we do is to provide kinetics of deactivation. So on the y-axis here, I'm plotting the surviving fraction, so how many spores survive versus the controls. And on the x-axis, I'm plotting here the fluence, the flux overlap multiplied by the exposure time. So this is what it looks like for a series done at 100 Kelvin. So you see if you have, so here just regular, just the controls, here no irradiation, everybody survives. And then the more you irradiate, the more photons you send, the more degradation you have. We did that for different temperatures. From room temperature, which a lot of literature has been done on. We also did some experiments at 200 Kelvin, which are temperatures that are relevant to, say, Mars. There's been some experiments that have been done for that as well. But this region, this temperature regions under that has not been explored, and that's the one that is relevant to the surface of ocean worlds. What's interesting here, just at first look, is that we do have kind of a tailing that that happens at the higher end. It could be due to the fact that we're very close to our sterility level. There's always some potential microorganism that can grow on our petri dish. So we have a sterility level here, confidence level. But we also see that we could also have some potential subpopulation that could survive, or maybe some clumping effects with the way we deposited, although that's something that I try to look at later, and I'm going to show you some results of why I don't think this is the case. But the main takeout point from this plot is that the spore viability increases as the temperature decreases. So if you compare the results for room temperatures, the brown data points here, if you compare that to what we have at low temperature, we can have orders of magnitude difference in terms of viability. That's quite interesting, and that's really reinforcing the data point that has been seen in the literature several years ago. I had some fun with these rates because I'm a physical chemist, so I decided to plot these rates versus the inverse of the temperature. And it does look like we have an Arrhenius-like behavior for these deactivation rates down to 60 Kelvin. So it's very similar to what we observe for chemical reactions. We see different results at very low temperatures. So in the case of chemical reaction, this could be explained by tunneling effects and so on. I don't want to say that we're looking at just a single chemical reaction. That might have a lot of different processes at stake here, of course, but it's interesting to see that we observe these trends, and that we could at a lot of potential chemical reactions occurring at once. So what do we think that is happening? Well, a lot of this irradiation experiments with UV photons has been looked at already at room temperature. There's been a really great series of, really body of work that has been done before, and the explanation here is that the UVs are actually creating damages at the DNA, breaking the double strand, and this is what we think could be explaining the deactivation rates that we're seeing. we're also reinforcing this hypothesis by looking at the spore themselves after irradiation. So these are some SEM images of the spore themselves, and they look pretty much similar to what we've seen, to what they look like before irradiation. So no morphological changes have been seen in our experiments for the doses that we've been using. So a little bit about the deposition technique that we use, since it's going to be important and may affect the results. So we used to deposit the spore monolayers a filter, membrane filter. So we're filtering some liquid with the spores in them, creating a wet surface on these membranes, inverting them onto our taps that we're going to put in the vacuum chamber. We've been pretty good at reproducing these nice layers, and as you can see from just simple phase contrast microscopy and also the ASCM images, it looks like we're really in the submonolayer regime, and that's very important because if you do the same work, but this time looking at droplets experiments, you're going to form potential coffee rings here. Just like when you dry a liquid, you can see these rings occurring on the surface. And this is definitely what we see for the case of spores. And what I did here was to do a different deposition with different amount of spores. So if you can see on the x-axis, and on the y-axis, you see the survival fraction are irradiated for, what was it, 100 joule per square meter here. And if you compare the result from the droplet experiments versus the deposition with the membrane filter that I see you on the right, that I show you on the right here, you can see that the viability rates are really different when you look at the droplets. So really something to look into, and this is something that is obviously really known in the planetary protection field and in other field where they look at decontamination processes. They've been actually having a way more fancier deposition technique than us, looking at spray deposition that can make really nice, even nicely spaced out spore layers compared to drop deposition. So again, we did this control here to make sure that clumping was not too much of a problem for our experiments. So now let's go back to what we're interested in, and it's really try to extrapolate these results to the case of to space and the surface of the icy moons. Can we do it? Can we go from the lab to getting some results in terms of viability for spores that would be at the surface of an ocean world? And the answer is yes. How do we do that? Well, the idea would be to first make sure that the photons that are inducing the damages are pretty much of the same efficiency for the lab that we have in the lab with respect to what we think we're going to have at the surface of an ocean world. So I'm showing you here the solar UV spectrum, what it would look like. So it's just the solar UV spectrum versus wavelength here, but at a distance far away from the sun, so at Europa in this case. Here is the spectrum of our lamp, kind of a black body. It's sort of the same. And here is the action spectrum for bacterial spore as was measured by Munakata et al. In 1991. The experiments that were done at the synchrotron when they irradiated Bacillus subtilis, actually, so the same bacterials, the same underspore that we've been using at specific wavelength. And by convolving these action spectrum with the two sources of photons, you can get an idea of what photons are the most efficient at deactivating the spores. And the profiles are pretty much similar between the lamp in our lab and that of the sun. We do have a little bit here contribution, but most of the damages would be inflicted by photons above 200 nanometers. So that's why we allowed ourselves to do this extrapolation. of our lab results to what we think would be going on in space. We have about a factor of 7 between the efficiency of our lamp and what we would have at Europa, and a factor of 24 for the case of Enceladus. And so when we use these results and the kinetic plots, we find that about 99% of the spores, if in direct sunlight at the surface of an ocean world, would be deactivated in about less than 10 minutes at Europa and less than 30 minutes at Enceladus. It's really, really fast. And I told you earlier that the low temperature were allowing us to get a couple of orders of magnitude in terms of viability. Well, it's not enough to catch these very long time scales that potential microorganisms may experience would they be at the surface of an ocean world. So viability, it's gonna be hard. We have potential ways to also, I mean, maybe this is the case that I really showed you for what if these microorganisms would be in direct sunlight. But most of these microorganisms may actually be found in ice. So here we did an experiment adding a layer of water ice on top of the spores, about microns. Actually was done by Aaron Noel in a previous paper. And what Aaron saw is that the addition of water, so comparing the navy blue markers here compared to the empty blue circles here, what Aaron saw is that there was no difference. Water was not doing much in terms of preserving these spores. This is about 1 micron of water ice here. And well, it makes sense because water ice actually does not absorb these photons, these efficient photons above 200 nanometers that I showed you earlier were the ones creating the damages to the spores. So that's an interesting result. Of course, if you add way more water there, it's not going to be the water itself, but scattering due to potential defect in the ice that may filter some of these UV photons. But you're going to have to go to millimeter or actually centimeter scales to have an efficient filtering. What could filter these photons is actually other components, other molecule components of the ice. And I'm showing you here the case of SO2. So SO2 is molecules that has been observed at the surface of Europa. We know it's present there. And the good thing with SO2 compared to water here, I'm showing you the plots of their absorption in ice. So here's water where you have very little absorption until you reach 160 nanometers. But if you look at the SO2 absorption cross sections above 200 nanometer for SO2, you have really strong electronic absorption. So what we did just to verify that physics was working is to add a layer of SO2 onto the spores. We compare it to, we compare the viability for a specific amount of fluence with and without SO2 ice, and we saw that viability was extremely increased because we had this shielding effect. So it's something to think about when think about when we're looking and trying to see what to expect in terms of viability at the surface of these worlds, that the potential additional components in the ice may shield microorganisms from harmful photons. So that's okay for the search for life. For planetary protection is good because you're going to need a lot of this material still to you're going to need a lot of this material to shield these microorganisms. So mostly what we're going to send there, if we have something saying there, will likely be deactivated very quickly by the solar photons. All right, so we continue this series of experiments, this time irradiating our bacterial spores with electrons. So we replaced the lamp by a flood electron gun. We stayed with energies under 10 keV for mostly safety reasons. Above 10 keV, we were creating a lot of X-rays. Safety was not really happy, so we decided to stay under this energy. And we followed the same process by depositing the spores, adding them into the tip of the cryostat, cooling to a specific temperature, and irradiating for a specific amount of time. And then we retrieved and counted the survivors. For these experiments, though, what we saw is that the substrate that we had a huge impact on our results. And well, of course, if you send electrons into a metallic surface, you're going to create X-rays. And these X-rays, we see them. Actually, we stick an X-ray spectrometer in our chamber. And what we did to avoid the effect of this X-ray was to add layers of ice. Layers of ice because it's relevant to what we're going to see. I mean, what we're looking at for the surface of ocean worlds, but also it allowed us to really dampen these X-ray lines coming from copper, but also the stainless steel that we're looking at. We needed to make sure that the electrons were still penetrating. So we needed to have an ice film that was not too thin, but also not too thick, so that the electrons would get to the spores. And so for us, for 3 keV electrons, in this particular case, our sweet spot was about 375 nanometers of water ice. So that's what we used. And we irradiated our spores, did that at multiple doses of electrons, and found that it takes quite a large dose of these electrons to deactivate the spores, 1.2 to the 14th keV per square centimeters to get about 99% of deactivations. So that's the result that we get for 3 keV. That's so we're right now, most of these experiments, when we irradiate material with particles of the specific energies, it's actually the secondary particles generated from this electron irradiation in this case that are actually doing a lot of the work. So we're working in terms of dose deposited at the material. So that's why most of the results performed at the specific energy can be extrapolated to higher energy. This is something that has been verified for the case of chemicals. This is not something that has been verified for microorganisms yet. This is not something that I could do at the moment, but I know of teams that are looking at this with higher energy sources of electrons, and hopefully we're going to have some data points to see if we can do this extrapolation. But assuming that we can, and things are working like for chemicals, what we found looking at the dose deposited at different spots, so here in the case of Europa, is that it would take about a month to have 99% deactivations. When we have here, we're about a centimeter down deep in the ice, and it takes about 10 years when we're at tens of centimeters. Still, when you look at geological timescales at these accessible sampling depth, these microorganisms will be deactivated, and will be deactivated pretty quickly. So where are we at now? What are the new directions that we're taking? So the first one, I told you when we were doing the UV work that we looked at these bacterial spores after irradiation using SCM, and we didn't see much damage, which was pretty interesting. We're using mild doses of radiation, but we continued this work using longer and higher radiation doses. And to see if the morphology of the spores could be, or microorganisms in general, could be a potential way to search for these microorganisms at the surface of ocean world, should they make their way up to the surface from a potential subsurface ocean. So what we did was to irradiate these pores in ice with different irradiation source for a very long time. So this is work that is performed by Lena Vincent. And yes, these morphological signatures seem to endure even for very large doses. So that's work that Lena presented like a month ago now at APSACON, and she's writing a paper on that. So stay tuned to look at that particular aspect. But the big question now, of course, and as a chemist, is something that I'm very interested in. What's happening to chemical signatures of these microorganisms when they are subjected to long irradiations? Will they stay there, and are there potential biotic versus abiotic signatures of their distribution? For example, here I'm showing you work that we're starting looking at fatty acids, mostly composing the coat of microorganisms, and trying to see if we can have differences before and after irradiation. It's proven to be difficult because extracting some of these chemicals takes some protocols that still need to be tuned. But it's something that we're working on in the moment. So fatty acids, we're also looking at proteins. It's going to be very interesting, amino acids and DNA. So that's the current work that we're doing, and we hope to be making some progress on that in the coming years. So now the other direction that I am taking myself, again, I'm not a biologist, so I went back to chemicals and to irradiation of ice, which is what I'm mostly doing. And the truth is that these laboratory experiments are done at a specific conditions under specific environments, but of course we're going to have many processes occurring at once when these microorganisms or even the signatures, chemical signatures of microorganisms are present at the surface of these worlds when they are subjected to radiation or to degradation rates. So what no one experiment is going to be able to reproduce all of these aspects, and is going to be able to do that for geological timescales. And that's where models are going to be very crucial into helping us figure out what to expect if we would send at some point a potential landed mission there. And so I've been trying to develop a model to look at these processes and get some ideas of what to expect. And I've been developing one model, especially in the case of Enceladus. So why Enceladus? Well, it's a very compelling target to like in general in the solar system for exploration. Any reasons? It's a very small moon. It's very bright. A lot of questions that we still don't know regarding of its physics. But of course, to me, the main interest of this target is potential at harboring life. We know it's a very active world with some plumes jetting that are actually feeding the earring of Saturn. And the flyby from Cassini was really outstanding and got us a lot of information on the chemical composition of these ice grains that are jetting out through the plumes of Enceladus, with a lot of interesting chemicals that are going to be very interesting for supporting life, and could some of them be potential signs of life that is being developed at the bottom of the subsurface ocean. So it's not just me who think that these are great targets. There are some concepts from missions that are being developed, and for instance, the Enceladus Orbilander has been put as one of the potential flagship mission by the recent Planetary Science and Astrobiology Decadal Survey here in the US. Enceladus is also part of the set of targets for the upcoming New Frontier mission calls. And also in Europe, a couple of months ago, I saw the announcement that Enceladus was the top target for the upcoming large class planetary mission that ESA would like to develop with the Voyage 2050 plan. So really great, great compelling target, but we need to start the work already at this point to prepare for the exploration of this body. We don't want to leave that for too late in the process, because there's going to be a lot of things that need to happen in terms of mission architecture, where to land, what instruments to bring in there. So having an idea of what to expect there is going to be paramount. So in this model that I've developed, there are three main components. And the first is that it integrates laboratory data in terms of degradation rates for a potential biosignature, or even for viability, as I showed you in the first part of the talk. And here, the idea for Enceladus is that UV photons are going to be the main source of degradation for these large, likely large for biosignature molecules there. And I showed you that for spores, it was photons above 200 nanometers that were doing the job. So what we did here in this part of the model is that we couple photolysis spectra of potential biosignature. I picked, for example, glycine and phenylalanine. Experiments have been done previously by Paul Johnson, actually, in our team. Also, a lot of literature from the astrochemistry field on irradiating ice with these large molecules and deriving photolysis rates. This is the plot that I'm showing you here. And in this model, we actually derive photolysis rates for a specific microorganism with respect to the amount of solar photons that you would have there. And what is interesting is that this effective photolysis rate is going to be highly depending on the type of molecules that you have, because of course each molecule is different electronic level, different absorption thresholds. And for example, here I'm showing you the convoluted photolysis, what I call photolysis efficiency for glycine versus phenylalanine. And what you can see is that if you integrate these photons, you have way less photons, way less degradation for glycine than you would have for phenylalanine. So these rates are going to be dependent, and yeah, each molecules, each species, components should be treated differently and individually. The second thing in the model is the fact that the UV flux will change at different location of Enceladus. So, and will also change versus ice depth. So if you look at how irradiation by UV photons would process at the surface of Enceladus, where you have Saturn going around the sun, at the same time you have Enceladus orbiting Saturn. And Enceladus is interesting because it's tidally locked. It's always the same face that faces Saturn. So you're going to have eclipse. Half of the time you're going to lose your photons. So you're going to have to divide your amount of solar photons by 2. And also Enceladus is slightly tilted. It's about 3-degree tilt, which means that in some area you would have really long periods of darkness that would happen. So for the longitude side, this is pretty much similar. The 3 degrees still doesn't do much, but if you look at the photons coming at different latitude here, this is what I'm showing in this plot. So starting from a Saturn year, summer, we have most of your photons, but in the winter, you're gonna have, you can even have for some of the higher latitude, for example, ADC, complete darkness period. So this is something that is gonna be important to look at when you're looking at the preservations of potential biosignatures. And the second thing here for the UV photons is that the profile of these UV photons will change versus ice depth, because Enceladus, it's very bright, it's a lot of water, but there's still some amounts of what has been called darkening material. There was a great paper by Amanda Hendricks who used HST observations and found that perhaps about 1% of the material of these ice grains could be actually pretty absorbing. And so when photons penetrate the top of the ice surface at Enceladus, some of them will be filtered by these darkening materials. So in this model, I'm integrating some optical constants. I chose those of tolerance here because it's the ones that we have. But of course, there are other material in there that could be filtering some of these UV photons, and it's something that we can integrate. So if your biosignature, potential biosignature is at a specific depth within the ice, it's not going to receive the same amount of photons. It's also not going to receive the same profile of photons. So that's something that the model integrates. And yeah, so here I'm showing you what's happening for a lot of darkening materials. It goes really, really fast. We're looking at micrometers of ice here and a loss of the UV flux by already orders of magnitude. So it's something to really keep in mind. And the final thing in the model is the fact that the material that is at the surface is actually not going to be there for a specific amount of time because you have a lot of plume redeposition on your surface. So that's, we've used some results from the paper from Southworth et al. That showed us here the redeposition rates per year at different locations of Enceladus. That's the map that you have here. Here you can recognize the vents coming from the tiger stripes that really redeposit some of the icy grains onto the surface. So we use all of this, put it in the mixer, and what we saw is that for Saturn year, this is what we have here, starting from 0 to year 29, actually, for Saturn year, you're going to have different depth profile for the survivability of these potential biosignatures. So when you start in the summer here, you're going to see a lot of degradation, but when you're going to go into the winter, you're going to have preservation, and then you're going to go back into the summer with some degradation again. So these are the parameters that I have. So it's phenylalanine for 6 micron diameter grains, and here we are at 60-degree latitude. So that's what we start with. We compared the preservation for different amounts of darkening materials. Of course, the more darkening material you have, the more preservation you have. Here I'm showing you the overall average preservation over Saturn year for the depth of the ice. We go up to 90% when we have 10% of darkening material. And here's what we have for different redeposition rates. And of course, the faster the deposition, the less time you have for your molecules to be photolyzed, so the more preservation you're going to have. What is interesting also to me from a physical chemistry point of view is the difference between different biosignatures. And here it's going back to the absorption profile of the different molecules. Glycine, that doesn't absorb as much of the solar UV photons, will have a higher preservation rate than phenylalanine. I also did the fun exercise of what's going on with spore viability. But here, as I told you earlier, things are deactivated pretty fast. In addition to getting depth profile, the model can also output maps of what we have on Enceladus. So it's these percentage that I showed you, but really here average over the map for different redeposition rates. And of course, the closer you are to the vents, the stronger the preservation the stronger chances you will have to retrieve some of the material from the subsurface ocean that is coming out from the plumes. But what's interesting is that, yeah, there are differences between two different signatures. This is glycine, this is phenylalanine. So it's something to think about if you're going to go sample at different location. Think about the fact that, yeah, different signatures are going to be there at the different level compared to how much you had initially in the ocean. So the recommendation based on this model is to go to locations where you have a higher deposition rates, above 0.03 millimeter redeposition per year, so to go to lower latitude. When I'm saying that to engineers, they're not super happy because, of course, the closer you are to the vent, the more scary it is for your payload. So you don't want to get too close either. But this is the kind of model that can give you some bracketing information for where to go if you land. But the main take-out message from this part is really that laboratory experiments are crucially needed for that. I showed you some example with specific molecules and specific darkening material optical constants, but we need this data. These are the only data that I could find. So doing these measurements for optical constant for typical Enceladus ice mixture would be very, very useful. And finally, we need a lot more work on wavelength-dependent photolysis rates for these big molecules. It's going to be important because the solar UV photons are going to be more harmful there for these big molecules, even compared to charged particles, because of the difference amount that you have. So we're really in dire need of that. All right, so that brings me to my take-home messages. So I showed you the work that we're doing to help with the search for life on icy ocean worlds, some experiments on the deactivation of bacterial spore, and specificity here is that we're doing it in ice at temperatures that are relevant to ocean worlds. It's you observe a slowdown for the case of UV irradiation, but things are going to be deactivated pretty fast. So viability may not be the best way that we have in these regions that are highly exposed to radiation to search for life. We're now exploring what's going on for longer irradiation in terms of cellular components, both in terms of morphology, but also to what's happening to the chemicals themselves. And finally, we're developing some models to help with mission concepts to help figure out what to expect there and to guide the development of future mission concepts, where to go if we want to maximize the science, if at some point we end up landing there. So, but more fundamental data are needed. And so that's really the takeout point of the talk. Let's do more laboratory work, let's do more irradiation work and more spectroscopy work. All right, so I'd like to thank the collaborators who worked with me on this project, funding agencies, the Habitable World Exobiology program, but also the Spontaneous Research Concept program at JPL. And many thanks to the EAI for letting me present my work here. And finally, thank you for your attention.
Yes, thank you, Edith, for this very interesting talk, and particularly also having a certain clue about some stability of molecules. The first part I had some problems to understand because it is well known that the surface is not very hostile, and it's really problematic for life forms. Therefore, for viability of cells, I never expect this. But on the second part, this was the main interesting part. I still have a question to you because you mentioned a number of isolated molecules you are testing, but you just imagine if we want to search for life, we have to search for cells. And this means this is a complex mixture of molecules. And this could be another story and look even different compared to what we are then analyzing. So I would suggest also in future not only using these isolated molecules, but a mixture of these molecules, even maybe also cell components, and to see about the stability under these conditions. This is always needed. We are also doing it. So therefore, I'm always saying also, a lot of groups have to concentrate on this because cells are more complex as just a few molecules, which are even organics in the interstellar medium. So please keep this in mind.
Absolutely. And yes, that's again, these are difficult experiments to do, though. We're trying to do them with fatty acids at the time. That's the first one we're starting. But yeah, extracting these components from the cell themselves, we're using subcritical water extraction at the moment, beating for the spores too. But getting a quantitative one-to-one from where we started to what we're analyzing is right now very challenging. So that's the point number one. But we have a good setup for the irradiation part. So I'm hopeful, and also the comparison between just pure like molecules themselves and the cell is going to be interesting. So yeah, I'm really with you on that, and I'm gonna look forward to results from your group as well, because that's something that we can absolutely feed into the model.
Yeah, they cross fingers because we are also doing a lot of investigations on the ISS, simulating this UV radiation and radiation impact on such kind of simulating samples. So this is something where we have to come together.
Absolutely. And yeah, it's going to be interesting, but yeah, looking at what the different subcomponents, how they filter the UVs and how they can shield some of the more inner
Yeah.
materials, the DNA and so on. That's a complex problem, but yeah, that's some cool physical chemistry
Cross
down there.
fingers.
Yes.
Thank you.
Thank you.
Jean-Hugues Filion, please go ahead.
Hi, Edith.
Hi, Jean-Hugues.
Thank you very much for this talk. Very interesting. I have a question about the experiment on the UV radiation, where you show that the temperature below 60K, then you have an effect that you have an increase over survival time. Do you have any hypothesis to explain this behavior?
So that's why I did the fun trick with the Arrhenius law. I just wanted to know if it was just the slowdown of the chemical reactions themselves that would explain why you have this increase of survivability. I think what would be interesting to do is to redo these experiments at wavelength dependent irradiation, but at low temperature to see if we can recover the action spectrum. And for the case of the room temperature experiments, it was seen that the action spectrum look a lot like that of the DNA. So at low temperature, it would be interesting if we follow that as well. So if it's just the same thing and it's just that chemical reaction are just slower, or if there's something going on, if there's another mechanism. There's also a lot of biology itself that can be used to see if we're actually looking at the same process of the DNA strand break and the proteins that do not keep the DNA together. For what's happening under 60K, that's another box can of worms here. And yeah, I think if we're looking at things happening like chemical reaction, I would think that maybe tunneling is happening, and that's why you're losing this dependence with the inverse of the temperature. But I don't know.
Okay, thanks.
Okay, so there were several questions in the chat, so I guess Jean-Pierre, you asked them, and also Jean-Hugues, you also asked your question, but there is one from Danielle, which is, what role does the surface play in your experiment? It seems to look like a copper substrate. Seems that you will elevate some electrons as well with the EUV.
Yeah, so that's a good question. So so we're using stainless steel for our taps for where we deposited the spores. So stainless steel, with the UVs that we use, they are actually lower energy. We're not looking at element alpha, so we're below the work function of the metal. So we're not going to create these electrons, I think. So that's the hypothesis now. But I agree that in the future, I hope it would be great to find a way to irradiate ice grains instead of spores with a surface of ice in between. That would be a better experiment, of course. And for the case of the electron irradiation, that's going to be that would reassure me in terms of the viability numbers that we're getting. So, but yeah, not for the UV, no, I don't think that's going to be the case. But for the electrons, yes, that's something I worry about, and we're going into actual ice. Inclusion of microorganism in ice would be very interesting.